friends, today we will start with our new chapter, that is chapter number 12 for class plus 2, and the name of the chapter is Biotechnology and its applications. And in chapter number 11 we are already discussed about the principles, processes of biotechnology, and in that chapter we will discuss how biotechnology is useful in various fields for the welfare of the human beings, what are the various principles that are used in the field of biotechnology, what are the various tools that are used in the biotechnology. So today we will start with our new chapter, that is the biotechnology and its applications. What are the various applications that are used in the field of biotechnology? So the use of biology to produce the products and the technology for the welfare of the mankind is known as biotechnology. So this is the use of biology to develop technologies and products for the welfare of human beings is known as बायोटेक्नोलॉजी. तो ऐसी बायोलॉजी का ऐसा यूज जो स्पेशली किस चीज को डेवलप करता है, जो डेवलप करता है स्पेशली टेक्नोलॉजीज एंड प्रोडक्ट्स को, और वो टेक्नोलॉजीज एंड प्रोडक्ट्स जो होती हैं दैट आर यूज्ड फॉर द वेलफेयर ऑफ द ह्यूमन बीइंग्स एंड दैट इज नोन एज द बायोटेक्नोलॉजी. एंड it has various applications. A biotechnology जो है, it has various applications that are used in the field of diagnostics, medicines, as well as crop production, waste management, energy production, as well as genetically modified crops. So these are the various applications that are used in the different fields such as forensics, diagnostics, processed food, waste management, energy production, as well as genetically modified crops, etc. So this is the brief introductory part of the biotechnology. Next is a briefing of the different applications of the biotechnologies that are used in the various fields. So these are the biotech applications, different types of the biotech applications that are to be used. So this is the genetically modified crops, that is the planting mats and hybrids, micropropagation, biofertilizers and biopesticides, animal improvement, and that is directly related to the field of the agriculture. Whereas rDNA products, vaccines and diagnostics, monoclonal antibodies, stem cells, tissue specific delivery methods, they are directly related with the healthcare. Next, environment directly produce the soil and the water remediations, biosafety and genetically modified organisms, and in case of industries, enzymes, fermentation based products and food, biotech instruments and equipments, bioenergy and biofuel, bioinformatics and biomining. These are directly related with the field of the industry. So these are the different types of the applications that are used in the field of the biotechnology. Next is the biotechnological applications in agriculture. What are the various biotechnological applications that are used in the agriculture? Biotechnology has different applications in agriculture. So biotechnology जो है, it has different types of the applications that are used in the field of agriculture. It can be used in agrochemicals. अब ये जो biotechnology जो है, that has to be various applications in the field of agriculture, and these applications are used in agrochemicals, organic agriculture, and genetically engineered crop based agriculture, and it can be used to produce genetically modified organisms. So these biotechnological applications that are used in agriculture, it is also used to produce the genetically modified organisms. Genetically modified organisms, that is GMO, that is genetically modified organisms, can be obtained by alteration in their genetic material. तो जो genetically modified organisms हैं, they can be directly altered in their genetic material. उनको जो है हम कैसे obtain करते हैं? They can be obtained by altered in their genetic material. Different applications of the genetically modified organisms are. So different types of the applications that are used in the genetically modified organisms are Number one, production of crops which are resistant to abiotic factors such as heat, cold, etc. Pest resistant crops help to reduce post-harvest losses. Minerals can be used efficiently by the plants, and the last one, that is the food with enhanced nutritional value. So these are the various applications that are to be used by the genetically modified organisms. The most important एप्लीकेशन इन द फील्ड ऑफ बायोटेक्नोलॉजी, सबसे ज्यादा इंपॉर्टेंट जो हमारी एप्लीकेशन है दैट इज टू बी यूज्ड इन द फील्ड ऑफ बायोटेक्नोलॉजी इज द फॉर्मेशन ऑफ पेस्ट रेजिस्टेंट प्लांट। तो सबसे इंपॉर्टेंट जो हमारी एक एप्लीकेशन है जो कि यूज होती है इन द फील्ड ऑफ बायोटेक्नोलॉजी दैट इज द फॉर्मेशन ऑफ द पेस्ट रेजिस्टेंट प्लांट्स, जिसमें कि एक फॉर्मेशन होती है किस चीज की, पेस्ट रेजिस्टेंट प्लांट्स की। एंड बैसिलस थ्यूनिंजिनेसिस is a bacterium that produces a Bt toxin। जो हमारा Bacillus thuringiensis है वो स्पेशली एक ऐसा बैक्टीरियम है जो कि क्या प्रोड्यूस करता है, that produces the Bt toxin which has insecticidal activity। और ये जो Bt toxin है स्पेशली it has the insecticidal activity। Genes that code for Bt toxin, जो जीन कोड्स करेंगे Bt toxin को, can be isolated, उसको हम आइसोलेट कर सकते हैं and inserted into the plants to provide insect। और उसको हम इंसर्ट कर सकते हैं किसमें, प्लांट्स में इंसर्ट कर सकते हैं। और जब हम उसको प्लांट में इंसर्ट करेंगे आफ्टर दैट दे हैव टू बी प्रोवाइड इंसेक्ट और पेस्ट रेजिस्टेंस। So next, what do you mean by Bt toxin? Bacillus thuringiensis produces certain proteins। ये जो Bacillus thuringiensis है it has to be produced certain types of the proteins that can kill insects। और ये जो certain types के proteins होते हैं they have the capability to kill the insects such as lepidopterans, coleopterans and dilipterans। So these are the types of the insects that are to be killed by this Bt toxin and they produce insecticidal proteins। ये insecticidal proteins को भी produce करते हैं which will enter into the body of the insect। जब वो insect की body में enter करते हैं, जब जैसे वो insect की body में enter करेंगे will become active। उसके बाद क्या होते हैं, insect की body में जैसे वो enter करेंगे, enter करने के बाद they immediately become active। This toxin when binds to the midgut produces, जब ये toxin जो होगा वो bind करेगा midgut को, after that they produces causes cell to swell। अब जो cells होंगे they causes to be swell and eventually death of the insect। और जैसे ही cells जो होंगे वो swell करना शुरू कर देंगे, immediately उसके बाद जो होगी वो insect की death हो जाएगी। These Bt genes are isolated। ये जो Bt genes होते हैं these are to be isolated and inserted into the cotton plants। और उसके बाद उनको insert किया जाता है cotton plants में। Such cotton is known as Bt toxin। इस तरह के cotton को जो है वो क्या कहा जाता है, Bt cotton। The genes that codes for insecticidal proteins, ऐसे जीन जो कोड करते हैं insecticidal proteins को, that are known as CRY-AC and CRY-AC। So this is the general information regarding the Bt toxin। So this is the diagram of the Bt toxin। This is the Bt toxin and Bt toxin is a genetically modified cotton crop है Bt toxin। और Bt cotton जो है, not toxin but Bt cotton, it is a genetically modified cotton crop। It expresses an insecticidal protein। ये क्या express करता है, it expresses an insecticidal protein which is delivered, और उसको कहाँ delivered किया जाता है, from a soil bacterium। That is to be delivered from a soil bacterium and that bacterium is Bacillus thuringiensis bar Stachy। So this is the general information regarding the Bt toxin and this is the resemblance of the Bt cotton। Next, pest resistant plants। What do you mean by the term pest resistant plants? Various pests affect the plants which causes loss as well as decrease in the yield of the plant। तो various types के जो पेस्ट होते हैं जो कि क्या करते हैं, प्लांट्स को अफेक्ट करते हैं और जैसे वो प्लांट्स को अफेक्ट करेंगे which causes loss, उसके बाद वो क्या कॉज करेंगे, लॉस को कॉज करेंगे एज वेल एज डिक्रीज, उसके बाद या फिर डिक्रीज होगा इन द यील्ड ऑफ द प्लांट्स, प्लांट्स की यील्ड में जो होगा वो ऑटोमेटिकली क्या हो जाएगा, डिक्रीज हो जाएगा। अ नेमेटोड मेलिडोडाइजीन Incogenitia infects the roots of the tobacco plants and causes a decrease in the yield of the plant. तो जो एक nematode है हमारा Meliodigene and Incogenita that affects the root of the tobacco plants. ये जो nematode है ये special tobacco plants के roots को जो है वो infect करता है and causes a decrease in the yield of the plant. और स्पेशली उस प्लांट की जो यील्ड होगी वो ऑटोमेटिकली क्या हो जाएगी, डिक्रीज हो जाएगी। जैसे ही वो निमेटोड उस प्लांट्स के रूट्स को अफेक्ट करेगा तो उसके अफेक्शन से जो होगा ऑटोमेटिकली उस प्लांट की जो यील्ड होगी वो क्या हो जाएगी, डिक्रीज हो जाएगी। टू प्रिवेंट दिस, अगर इस चीज से इसको बचाना है, आरएनए इंटरफेरेंस टेक्नोलॉजी वाज यूज्ड। अगर इस चीज से हमें बचाना है कि प्लांट्स की यील्ड को डिक्रीज होने से बचाना है, तो दैट मींस एट दैट स्टेज the RNA is to be RNA interference technology जो है उसको क्या करना पड़ेगा, यूज करना पड़ेगा। एंड दिस मेथड इन्वॉल्व साइलेंसिंग ऑफ ए स्पेसिफिक मैसेंजर RNA, और ये जो मेथड है वो इन्वॉल्व करेगा साइलेंसिंग ऑफ द स्पेसिफिक मैसेंजर RNA ड्यू टू कॉम्प्लीमेंट्री GS RNA molecule. This inhibits the translation of the messenger RNA, और ये क्या करेगा, इनहिबिट करेगा translation of the messenger RNA. So this is the pest resistant plants. Next, this is the development of the pesticide resistant plants. How pesticide resistance dwells in the various types of the plants. So these are the pest eating plants, and that is the mite, and this mite especially eats the plants, and this is the pesticide applications that are to be used against such type of the plants. And this is the resistance individuals survive, how resistant individuals survive, and this is the resistant individual reproduce, how the resistant individuals they will be produced, and these are the pesticide applications, and this is the pest survive application. So these are the development of the pesticide resistant plants that are to be shown in this diagram. Next is biotechnology applications in medicines. After the plants, next our important application of the biotechnology is that is in the field of medicine. How biotechnology they have to play an important role in the field of medicines. So biotechnology has contributed a lot in the medicine industry, such as plants. Biotechnology has also played an important role in the field of medicine industry. The use of biotechnology in medicine is known as medicinal biotechnology. जो use हमारा biotechnology का medicines में हो रहा है, तो उस चीज को जो है वो हम क्या करेंगे, medicinal biotechnology. They provide methods for the formation of genetically modified insulin. अब ये जो हमारी एक एप्लीकेशन है बायोटेक्नोलॉजी की, दैट इज टू बी यूज्ड इन द मेडिसिन्स, ये क्या करेगी, ये जो होते हैं वो प्रोवाइड करते हैं, दे प्रोवाइड द मेथड्स, ये मेथड्स को प्रोवाइड करेंगे स्पेशली फॉर द जेनेटिकली मॉडिफाइड इंसुलिन, एंड दैट इंसुलिन इज नोन एज ह्यूमुलिन, एंड दैट इंसुलिन इज नोन एज ह्यूमुलिन, and this humulin helps it, अभी humulin जो होगा वो help करेगा in the treatment of large number of diabetes patients. This type of the humulin, that means it has to be play an important role or play an important role in the treatment of the diabetes patients. Earlier insulin was, पहले जो था वो insulin was extracted from the pancreas. Earlier जो insulin था that was to be extracted from the pancreas of the slaughtered animals such as cattle and pigs. तो सबसे पहले जो इंसुलिन था that is to be extracted from the insulin is to be extracted from the pancreas of the slaughtered animals, and these slaughtered animals are especially cattle and pigs. Such insulin has chances to cause allergies. ऐसे इंसुलिन के पास जो थे वो chances थे कि they have to be cause the allergies to humans, and the most important issue with the production of insulin is formation of mature and assembled insulin. तो सबसे important issue जो है हमारा with the production of insulin जो है that is the formation of the mature and the assembled insulin. Next is the human insulin production, that is to be shown with the help of this picture. How human insulin production takes place, that is to be clearly shown by this diagram. So this is the human pancreas cell, and in the human pancreas cell this is the DNA and these are the human insulin producing genes. ये human insulin जो है that are to be produce the genes, and this is the introduction of recombinant DNA into the bacterial cell. यहाँ पर जो हमारा recombinant DNA है that is directly introduced into the bacterial cell, and this is the recombinant bacterium. And after recombinant bacterium, this is the human insulin, and how human insulin, that is extraction and purification of the human insulin takes place. And with the help of this diagram, this is the fermentation tank, and in this fermentation tank recombinant bacteria multiply. इसमें जो हमारे recombinant bacteria हैं, and that recombinant bacteria are in this fermentation tank. So these recombinant bacteria multiplying, ये जो हमारे recombinant bacteria होंगे they continually में वो multiply करेंगे, and जैसे वो multiply करेंगे after the multiplication they produce human insulin. जैसे वो कंटिन्यूटी में मल्टीप्लाई करेंगे और मल्टीप्लाई करने के बाद प्रोड्यूस करेंगे ह्यूमन इंसुलिन को इन दिस फर्मेंटेशन टैंक. And after this fermentation tank, this is the formation of the human insulin, and this is the bacterial DNA that is to be present in the bacterium, and beside bacterial DNA this is the plasmid DNA. That means in the bacterium both bacterial DNA as well as plasmid DNA both are present in the bacterium, and this is the plasmid DNA cut with the restriction enzyme. अभी plasmid DNA जो होगा that is to be cut with the help of this restriction enzyme, and after that there is the formation of the recombinant DNA. So this is the human insulin production, how human insulin production takes place, that is to be clearly shown with the help of this diagram. Next is the gene therapy. This is the most important topic of this chapter, that is the biotechnology and its application. What do you mean by gene therapy? To correct the hereditary diseases, gene therapy is used. अगर hereditary diseases को correct करना है, that means the main concept or the main therapy that is to be used is the gene therapy. The correction of the gene defects is known as gene therapy. जो हमारी correction होगी जीन्स में, जो हमारी करेक्शन होगी जीन डिफेक्ट्स में, जितने भी हमारे जीन्स में डिफेक्ट्स होंगे और उन जीन डिफेक्ट्स को करेक्ट करने के लिए जो जिस चीज का ये जिस थेरेपी का हम यूज करेंगे, दैट थेरेपी इज नोन एज द जीन थेरेपी. अ नॉर्मल जीन इज इंसर्टेड इनटू द इंडिविजुअल. जो नॉर्मल जीन होगा दैट इज टू बी डायरेक्टली इंसर्टेड इनटू द इंडिविजुअल. उसको इंडिविजुअल में इंसर्ट किया जाएगा और इन एन एम्ब्रॉय, या तो उस नॉर्मल जीन को एक इंडिविजुअल में इंसर्ट किया जाएगा या फिर एक एम्ब्रॉय में इंसर्ट किया जाएगा टू रिप्लेस द एबनॉर्मल जीन. और उस नॉर्मल जीन को जो है इंसर्ट करने का इंडिविजुअल में या एम्ब्रॉय में जो इंसर्ट करने का जो लॉजिक है, दैट इज टू बी रिप्लेस द एबनॉर्मल जीन, ताकि वहां से जो एबनॉर्मल जीन जो है वो रिप्लेस हो सके. The first gene therapy was given to a, जो सबसे पहली जीन थेरेपी थी was given to a 4-year-old girl. जो सबसे पहली जीन थेरेपी जो थी that was given to the 4-year-old girl in 1990. 1990 में जो पहला हमारा जीन थेरेपी का जो कंसेप्ट था फर्स्ट जीन थेरेपी का वो दिया गया था 4-year-old गर्ल को in 1990 में for adenosine deaminase deficiency. और इस डिजीज के अगेंस्ट जो था उस फर्स्ट जीन थेरेपी को दिया गया था, दैट इज द adenosine deaminase deficiency. And earlier bone marrow transplantation, और earlier जो bone marrow transplantation है and enzyme replacement जो है therapy were used to treat adenosine deaminase deficiency. तो अर्लियर अगर हम पहले देखें तो जो बोन मैरो ट्रांसप्लांटेशन है या फिर एंजाइम रिप्लेसमेंट थेरेपी जो है, दे वर यूज्ड टू ट्रीट दिस डिजीज दैट इज द एडेनोसाइन डीमाइनेज डेफिशिएंसी. बट बोथ स्ट्रेटजीज आर नॉट कंप्लीटली करेक्टेड. तो दोनों जो हमारी स्ट्रेटजीज थी दे आर नॉट कंप्लीटली करेक्टेड. जो क्योर नहीं थी पूरी तरह से, दे आर नॉट कंप्लीटली टू बी क्यूरेटिव. So this is the gene therapy and this is the cell. that is with non-functioning gene. ये एक सेल है, that cell is with the non-functioning gene. अगर हम उसके ऊपर functioning gene जो है वो insert कर देते हैं किसी individual में या embryo में, after that the cells functioning normal. तो उसके बाद जो body के जो cells होंगे, that means these cells functioning normally. So this is the basic concept regarding the gene therapy. Next is molecular diagnosis. What are the various steps that are to be used against this disease, and that steps are to be in the form of molecular diagnosis. It helps in disease diagnosis by various techniques such as ELISA, PCR and recombinant DNA technology. तो ये जो molecular diagnosis है, इसमें जो है वो helps करते हैं disease diagnosis में by various techniques, और उन disease diagnosis के लिए various techniques का use किया जाता है, and these techniques are especially in the form of ELISA, PCR and recombinant DNA technology. ELISA, that is enzyme linked immunosorbent assay, that is the full form of the ELISA, is based on antigen and antibody reaction. और especially ELISA जो है technique जो है वो किसके ऊपर based करती है, that is based on the antigen and the antibody reactions to detect different diseases. अब जब ये किसके साथ एक interact करती है या based करती है, that is antigens and antibodies के ऊपर based करती है to detect the different diseases. PCR, that is a polymerase chain reaction, is a technique to amplify specific DNA segment, जो कि क्या करती है, amplify करती है या specific DNA segment को. That is in case of the PCR, that is a polymerase chain reaction. And this helps to detect HIV/AIDS patient, और especially ये जो technique है, this is specially used to detect the HIV virus in the AIDS patient. Next is transgenic animals. What do you mean by transgenic animals? Animals that have their DNA manipulated are known as transgenic animals. ऐसे animals जिनके पास की DNA manipulated है, manipulated DNA, that is known as the transgenic animals. For example, rats, rabbits, pigs, sheep, etc. These are the animals that they have their manipulated DNA. So these types of the animals are known as the transgenic animals. These animals helps to understand how genes are regulated. ये जो हमारे animals हैं, ये हमें इस चीज में हेल्प करते हैं, अंडरस्टैंड करने में हेल्प करते हैं कि हाउ दीज जीन्स आर रेगुलेटेड. ये जीन्स होते हैं वो किस तरह से रेगुलेट होते हैं, एंड हाउ दे हेल्प इन द डेवलपमेंट ऑफ एनिमल्स. एंड ये जो हमारे जीन्स हैं वो किस तरह से एनिमल्स की डेवलपमेंट में हेल्प करते हैं. It helps to study how genes are responsible for the development of the various diseases. साथ में ये हमें ये भी हेल्प करता है इस चीज की स्टडी करने में कि हाउ दीज जीन्स आर रिस्पॉन्सिबल, कि किस तरह से ये जीन्स जो हैं वो रिस्पॉन्सिबल हैं फॉर द डेवलपमेंट ऑफ द वेरियस डिजीज दैट आर टू बी अकर इन द डिफरेंट टाइप्स ऑफ द एनिमल्स. They can be used to manufacture chemicals. ये chemicals को manufacture करने में भी help करते हैं. That are useful, जो कि ये chemicals होते हैं किसी न किसी तरह से कहते हैं वो useful होते हैं. They can also be used for testing of drugs and vaccines. ये drugs और vaccines को test करने के लिए भी helpful है. So this is the picture of the transgenic animals and how these transgenic animals that are to be takes place, and these are the organisms to be cloned, and these are the 12 differentiated cells. These are the differentiated cells that taken from the donor, and these cells begins to develop as an embryo. ये cells होंगे they have to begins to develop in the form of an embryo, and these are the implanted into a eve, and this is the clone is born. यहाँ पे clone जो है वो born करेगा. These are the egg cells, and the cells to be cloned are fused together. इसमें egg cells होंगे या फिर cells जो होंगे they are to be fused and cloned, and after that this is the egg cell with no nucleus. ये egg cell है जहाँ पर कि कोई nucleus नहीं है, without nucleus. This is the egg cell, and this is the nucleus where the egg cell is to be removed. ये nucleus है जहाँ से कि egg cell को क्या किया गया, remove कर दिया गया है. And this is the unfertilized egg cell, और ये जो है this is the unfertilized egg cell. and this is the glass pipette that is smaller in diameter than here. और ये जो हमारी ये glass pipette है that is to be smaller in diameter than the here. This is the formation of the transgenic animals. How the formation of the transgenic animals takes place, that is to be clearly shown with the help of this diagram or this picture. Next is biotechnology in production of antibiotics. What is the important role that is to be played by the biotechnology in the production of antibiotics? So plants are used to develop antibiotics for humans as well as for animals use. तो plants जो होते हैं they have the capability to develop the antibiotics, and these antibiotics are both useful in case of the humans as well as in case of the animals. It helps in the production of antibiotics. ये जो है किस चीज की प्रोडक्शन में हेल्पफुल है? एंटीबायोटिक्स प्रोडक्शन में, वैक्सीन्स एंड आर्टिफिशियल हार्मोन्स फॉर हार्मोन्स थेरेपी, जो और आर्टिफिशियल हार्मोन्स को भी डेवलप करते हैं स्पेशली फॉर द हार्मोन्स थेरेपीज. नेक्स्ट इज एथिकल इश्यूज. व्हाट आर द वेरियस एथिकल इश्यूज? First ethical issue is the manipulation of the living organism by man is similar to man playing God. सबसे पहले जो हमारा manipulation है living organisms का by the man is just similar to the man playing God. It is a tool with great power. ये एक ऐसा tool है that is to be with the great power and therefore immense responsibility. और इस वजह से इसकी जो एक responsibility है that is to be immense responsibility. The genetic modification of any organism is not always completely predictable. जो genetic modification है किसी भी organism की, that is not to be always completely predictable. It may lead to problems. ये problems को भी lead कर सकता है in the organisms. The impact of introduction of genetically modified organisms into an ecosystem is not completely known. It could have unpredictable results. इसके कुछ unpredictable results भी हो सकते हैं. Next is GEAC. What do you mean by the term GEAC? That is Genetic Engineering Approval Committee. GEAC. The full form of the GEAC is the Genetic Engineering Approval Committee. Is a committee set up by the Indian government. ये एक ऐसी committee है that is to be set by the Indian government to oversee all decisions, जो कि क्या करती है? Oversee करती है all decisions को regarding GM research and the safety of the GMOs for the public use, regarding genetically modified research as well as the safety of the genetically modified organisms, that is for the public use. Next is biopiracy. This is the most important topic of this chapter, that is biopiracy. Use of bioresources by commercial and multinational companies and other organizations without the appropriate authorization and permission from the countries and people concerned without making the compensatory payment. तो जो हमारी बायोप्रेसी है इसमें यूज है बायोरिसोर्सेज का बाय कमर्शियल एंड मल्टीनेशनल कंपनीज एंड अदर ऑर्गेनाइजेशंस विदाउट द अप्रूवल ऑथराइजेशन, जिसमें कि कोई एप्रोप्रिएट ऑथराइजेशन नहीं है, एंड परमिशंस फ्रॉम द कंट्रीज, कोई परमिशन नहीं है कंट्री से, एंड पीपल्स कंसर्न विदाउट, और इसमें जो पीपल है कंसर्न विदाउट मेकिंग द compensatory payment. So this is the concept, that is the biopiracy, and this is all about this chapter.